Hello everyone, I am Nazim. I am studying as a PhD student in Department of Material Science and Engineering at Channam National University. We will examine bioelectrochemical fuel cell article which written by Lemuel B. Wingard, Jr. Ching Hao Shaw and James F. Kastner in Mr. Zhong Suki's lecture. We will try to find answers to the most frequently asked questions about bioelectrochemical cell. Let's start. We see contents in this study. At the beginning, we will see general information about fuel cell and then disadvantages of fuel cell. We try to understand the benefits of using bioelectrochemical fuel cell and what is the importance of these cells. Then, this topic is followed by microbiological and enzyme fuel cells. After that, we try to explain cofactor and what is the function and importance of cofactor. Finally, we will summarize studies in conclusion step and what is the research needs. A device which directly converts chemical energy to electrical energy is called as fuel cell, which consist of anode, cathode, electrolyte and external circuit. Some reactions occurs in anode and cathode. Let us consider reactions in fuel cell. In the anodic reaction, as a result of a metal is oxidized, releasing electrons into the metal. In contrast to anodic reaction, a solution species, often O2 or H+, is reduced, removing electrons from the metal in cathodic reactio. Fuel cells work like batteries, but they do not run down or need recharging. They produce electricity and heat as long as fuel is supplied. A fuel cell consists of a negative electrode, or anode, and a positive electrode, or cathode, sandwiched around an electrolyte. A fuel is fed to the anode, and air is fed to the cathode. In a hydrogen fuel cell, a catalyst at the anode separates hydrogen molecules into protons and electrons, which take different paths to the cathode. The electrons go through an external circuit, creating a flow of electricity. The protons migrate through the electrolyte to the cathode, where they unite with oxygen and the electrons to produce water and heat. After anode and cathode, the third part in fuel cell is electrolyte. There are three types of electrolyte. First, acid electrolyte helps hydrogen ions movement from anode to cathode, such as hydrogen bromide. Alkaline electrolyte allow hydroxide ion movement from cathode to anode. Finally, viscous type electrode, such as molten carbonate and solid oxide, help the ions migrate from the cathode to the anode. What is the effect of electrolyte type? Of course, it directly affects operation temperature. Let us discuss the efficiency of fuel cell now. The efficiency formula was originated from Gibbs free energy formula. In generally, T delta S equals 10% of delta H for many system. Hence, theoretical efficiency equals %90, but of course it is impossible in practical, because theoretical efficiency means there is no current flow. Generally, efficiency varies between 25 to 70% in practical, mostly 55 to 60%. Why efficiency decreases so much, compared as theoretical efficiency, sum of potential, E degree, are not available when currents start to flows. This unavailable part can be defined as overpotentials or polarization, with depend on system S kinetics. Let us see types of overpotentials. Generally, origination of overpotential types are depend on the value of current density. Activation, ohmic and concentration potential are shown as point A, B and C, respectively in the graph. Activation overpotential generated at very low current densities. Ohmic and concentration overpotentials generated high current densities where power of fuel cell is become zero by mass transfer resistances. Why we need to bioelectrochemical fuel cell? Although fuel cell has high operating efficiency, it has many disadvantages. Some of them, 1. The limited viability in high cost of the catalysts, platinum, nickel, etc., 2. The highly corrosive electrolytes, 3. The elevated operating temperatures, 4. Problems of electrode development, 5. Short operational life. These problems will be overcome by development of bioelectrochemical fuel cell. First let's see what is the bioelectrochemical fuel cell. Bioelectrochemical fuel cell can be defined as the use of enzymes or microorganisms as components of one of the fuel cell electrode compartments. Why we should consider it, instead of fuel cell? Because its advantages are so interesting. Let's we see figure, operation at room temperature, then much less corrosive electrolytes. Finally, high selectivity of enzyme catalysts. In addition to, overpotential problems can be reduced by using biological materials according to some studies, but this approach is still controversy. Now let's we consider microbiological fuel cell. One of the easiest way for constructing bioelectrochemical fuel cell is combination of inert solid electrode, such as platinum, and suspension of selected microbial cells. This combination leads anodic chamber of fuel cells. Metabolic activity of the cells affects potential between anode and cathode and current. For example, with increasing concentration of cell, the oxygen consumption at the anode increased because of aerobic fermentation. There are many difficult questions for bioelectrochemical fuel cells. One of them is determination of compatibility of substrate materials and microorganism. To initiate the initial reaction step at anode is difficult due to most hydrocarbons are not electrochemically active. Glucose is found very good candidate for this aim in different studies. According to some scientists, power output for these fuel cells depended on the concentrations of microbial cells and glucose as well as on the oxygen flux and binding affinity of the cells for the substrate. However, the relationship between these parameters are still controversy. Another one of the type is enzyme fuel cell. In contrast to microbial fuel cell, enzymes or enzyme cofactors are used in anodic for oxidation or cathodic electrode for reduction reactions. Now, please consider table. There are different types of enzyme and electron mediator types in there. Scientists try to find their effect on properties. Because each enzyme has different effect on fuel cell. Some of the results, firstly, to reduce solution viscosity and enhanced oxygen transport, hyaluronidase is so available. Secondly, as a result of glucose with platinum surface in contact with enzyme anode, current densities of 10 to 15 microampere divided by square centimeter produced. Thirdly, with the increasing concentration of mediator, anodic current increases. To summarization, the effect of enzyme types, concentration of mediator and effect of some parameters on current density could be found as a result of these studies. As a result of these studies, some parameters which affect electric current have found. Resulting current can be affected by 1. Concentration of enzyme and substrate, 2. Michaelis constant, 3. The rate constant for breakdown, 4. A ratio between reaction rate and transport rate. Why we need cofactor? The combination of cofactors with oxidoreductase enzymes act as electron and proton transfer agents. At the enzyme anode, electrons must be transferred readily from cofactor to the electrode surface. One of the aim of using cofactors is improve electron transfer problem. To improve electron transfer, one of the best way is enhancing cofactor electroactivity by cofactor immobilization. Let us we consider figure. The advantages of this structure, generation of orbital overlap from N1 and N5 through to the C8 bridge, thus affording a pathway for electron delocalization, because of its ability of electron conducting linkage to give a pathway for easy electron transfer from the reduced cofactor onto the electrode surface. Flavins was preferred. In this study, some theories of bioelectrochemical fuel cells and its common problems are described and determined information which should be need to improve it for practical purposes. Although fuel cell has high operating efficiency, it has many disadvantages. The possibility exists to reduce some of these problems through the development of bioelectrochemical fuel cells. There's still many controversy points for bioelectrochemical fuel cell because of deficiency of theoretical information. Because it is very new research area, compared as others. So they should be investigate for these purposes. Some of them are shown in figure. 1. Comparative electrochemical performance data. 2. where the microbial cells are undergoing aerobic metabolism. 3. Mediator candidates of high electroactivity in rapid rates of diffusion should be given in-depth study. 4. Fundamental work on microbial electrodes in addition to electron transfer mechanisms. 5. Rates of diffusional transport of NAD in mediators. I want to finish my presentation with one proverb. In science, the important thing is to modify and change one's ideas as science advances, by Herbert Spencer. That's why we should never say, why do we need a bioelectrochemical fuel cell when we have a fuel cell? Many opinions will change as scientists investigate the conflict points of the bioelectrochemical fuel cell and find new solutions to them.